drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. The full version of this episode is available to paid subscribers at the Mind and Matter Substack at mindandmatter.substack.com. The full version is also freely available on my YouTube page. Just look up Mind and Matter or my name, Nick Jikomis, on YouTube, and you'll find the episode there. This is a partial version of the episode, and so it'll cut off partway through, but you'll still get a significant portion. So sign up to become a paid subscriber at Substack or visit my YouTube page to get the full episode. By just telling everyone a little bit about who you are and what you study?
Yeah, my name is Nadal, as you said. Well, I really began to study chemistry in my hometown, which is Alicante in the southeast of Spain. And later I moved to the Autonomous University of Madrid to study biochemistry and molecular biology. And after that, I decided to come back to Alicante, where is an excellent institute of neuroscience, and I did my PhD in that institute. Although I work with beta cells that are not really neuron, but from an electrophysiological point of view, we were interested in studying the stimulus secretion coupling in pancreatic beta cells and how the electrical activity of the beta cells drive to the calcium signaling that produces the release of insulin in those cells. So it makes sense to study that, being in an institute of neuroscience. In the sense that we were very interested in ion channels and calcium signaling and those stuff. So I did my PhD there, and later I moved to King's College London, UK, to do my postdoc there. So I stayed in King's College 4 years, and I was studying the connection between calcium signaling and cell division in astrocytes. So I was really in a group of neuroscientists and working particularly on that, how proteins such as albumin that were going through the blood-brain barrier, what the blood-brain barrier breaks down, activates astrocytes and make them to divide and produce a scar, and this sort of stuff. And really it was in London when I first learned about endocrine disruptors. That was at the mid-90s. And also about the extranuclear actions of estrogen receptors. So at the end of the 90s, I moved to Alicante because they opened a new university here, and they contact me to go back, let's say, home and start in this new university. And I decided to begin working in estrogens and endocrine disruptors in the pancreas, in the endocrine pancreas, the beta cells. So that's more or less my background.
No, it's an interesting background. And, even though we're going to talk probably mostly about the pancreas and, endocrine aspects of the pancreas, hormone secretion, insulin, estrogens, things like this, and endocrine disruptors, I think it's worth reminding people that we'll probably talk about the brain to some extent at some point, maybe. But even though we think about like the pancreas as an endocrine organ, and we don't think of it like a neural organ because it's obviously very different from the brain. At the same time, much of the brain, important parts of the brain that we might talk about, are neuroendocrine organs themselves. They also secrete hormones, and even though the pancreas isn't made out of neurons like the brain is, part of your studies were studying the
Mhm.
electrical properties, ion channels, all these things that we normally associate with neurons are found in other cells as well.
Exactly. Well, I think I consider myself a beta signaler. And so the beta cell works very much as a neuron from that point of view,
Yeah.
that
Yeah.
it's an excitable cell, it produces action potential, and are these action potentials which code finally for the insulin release,
Mm.
very similar to what a neuron does uh, for neurotransmitter release.
Yeah. So yeah, let's talk about the pancreas a little bit just to give people some sense. You're referring to the beta cells. So these are a major cell class, if I understand correctly, they're a major cell class of the pancreas, and these are the ones that secrete insulin. You're actually saying that analogous to the way that a neuron will release neurotransmitters in response to a change
Mhm.
in electrical signaling within the cell, something similar is happening to regulate insulin release from these beta cells in the pancreas.
Mhm. Yes, the pancreas, we have two types of pancreas, the exocrine pancreas, which is not related uh, um, to the endocrine system, and the endocrine pancreas, which are islet of Langerhans are called, which is the basic physiological unit of the islet of the endocrine pancreas. And we have in humans million of them inside the pancreas.
Mhm.
So each of these islets contains between 1,500 to 3,000 cells of five different types. Uh, the majority of those five type of cells is the pancreatic beta cell. In human, it's about 50 to 60% of the islets are pancreatic beta cell, and about 40% are glucagon releasing alpha cells, and the rest are minority somatostatin releasing cells and others. In mice, it's a little bit different. They contain more beta cells, so about 80% of the cells in the islet are beta cells, and they contain less alpha cells releasing glucagon, about 10%, 15%.
Mhm.
So that makes a bit of a change when we compare the situation in mice to the situation in humans.
I see. So one portion of the pancreas is devoted mostly to endocrine functions. So this is the one that
Yeah,
people probably hear about most that's associated with insulin and blood glucose regulation. Basically, you're saying that chunk of the pancreas is composed of what we call islets. So these are basically patches of tissue, each of which contains a certain combination of cells, such as the insulin secreting cells and others.
exactly. Yes.
And so I don't want to spend too much time on this because I've covered it on the podcast before. And I think people are most familiar with this part of the pancreas. And we're going to also talk about other things the pancreas does that I think people don't appreciate as much. But
Mhm.
can you give us just a very basic, very simple rundown of how these beta cells and cells in the endocrine portion of the pancreas regulate blood glucose levels via insulin?
Yes, the pancreatic beta cell is the cell that produces insulin. It expresses insulin and biosynthesizes insulin, and is the only cell really that is able to release this insulin in response to nutrients
Mm-hmm.
in our body. And that's very important because insulin is the only hormone able to decrease blood glucose.
The only one we
So
know about.
the only one we know about. So if the pancreatic beta cell goes wrong,
Yeah.
then we get diabetes.
Yep.
And that's why it's very important for all of us. Um, uh, so normally after a meal, what we have is an increase in blood glucose. And this is because well, the digestion, and there is, of course, increase in glucose, amino acid, and fatty acids, and they go to the pancreatic beta cell. So the pancreatic beta cell, if we focus in glucose, uptakes glucose, metabolize glucose through glycolysis and the Krebs cycle, and produces an increase in ATP-ADP ratio. And this connects to a channel at the plasma membrane, which is the ATP-sensitive potassium channel, that controls the resting membrane potential of the cell. So when the ATP increases, this channel closes, potassium stops going out of the cell, and then the plasma membrane depolarizes, opens calcium channels, voltage-gated calcium channels, and calcium gets in the cell and produces the exocytosis of insulin. And then insulin is released into the blood.
Yeah, and this is like what you were talking about before. It really is a lot like neuronal transmission in the brain in many ways. There's ion channels that are coupled to ATP, ADP levels in the cell. So, metabolism is being coupled to channels that regulate the potential of the cell. And when the cell becomes sufficiently excited, certain ions come into this. So there's like a more abundant ATP. This causes things like potassium and calcium to flow back and forth between the cell. And at a sufficiently altered membrane, the cell will then release, in this case, insulin, not unlike the way that a cell in the brain might release neurotransmitters.
Yeah, exactly. That's it.
Interesting.
That's it.
Okay, so insulin gets released, and basically it's telling the body, take some of that glucose out of the bloodstream and store it in cells.
Exactly. Insulin has receptors in different tissues, but mainly it works at the liver, skeletal muscle, the adipocytes, and also in the brain.
Mm-hmm.
So what insulin says to the skeletal muscle, please uptake glucose, and indeed is the skeletal muscle, the tissue that takes more glucose away from our blood, tells the adipocytes also to uptake glucose, and the excess of glucose is stored, it produces lipogenesis, and it tells the liver to stop producing glucose, which is the function of the liver in terms of glucose homeostasis. And it also tells the brain that insulin has been released and there is nutrients already there,
Yeah. So it's
not
serving
more food intake. Yeah.
Yeah. So the insulin's not only telling parts of the body, like the liver, to store the glucose and basically save it for later, but it's also telling the brain, like, basically that we just ate. It's important for instigating some of the satiety regulating mechanisms of the brain. It's saying we just ate something. There's nutrients. Prepare the organism to deal with that.
That's it.
And so this is a core mechanism of the body. It's super important that blood glucose levels don't get too high or too low. Insulin secreted from the pancreas. You're saying the pancreas is these are the only known cells in the body that secrete insulin. So it's extremely important in that sense. What is so important about preventing glucose levels from getting too high for too long? What are some of the things that start to go wrong if glucose remains elevated?
Yeah, if glucose remains elevated, it starts to affect mainly endothelial cells and the vascular system, and that begins to produce problems. And this is common. I'm not an MD, but I know a little about that, and it is common in diabetic people for many years to have complications with in the eye, for instance, the retina, and to have problems of vision, to have diabetic foot because of this lack of microvascular tissue, which is affected there. It affects the heart and makes you more have a higher probability of a heart attack
Hmm.
and other things. So really having high levels of glucose for uh, a long period of time is nasty. So is to our organism.
Yeah, yeah.
So
And I mean, on a sort of coarse-grained, intuitive level, I would imagine, glucose is fuel for the cell. It's like a primary fuel
yeah,
source for all of our
yeah.
cells. You don't want to give them too much too often. I guess, maybe an analogy would be like, I have my laptop here and it's plugged into the power source, but the power source has a specific voltage. It's sending a specific amount of power. If I was to hook this up to a massive generator that would send too much power all at once, I could
Mhm.
blow out some of the circuits of my computer.
Yeah. Yeah, that's it. I mean, the point is we always need to regulate glucose very accurately, and the beta cell is prepared for that.
Mhm.
So it's prepared to have a response in terms of insulin release to very small amount of insulin. It can distinguish between 6 millimolar to 6.1 millimolar,
I see. It's highly sensitive.
6.2 millimolar, very highly sensitive to those changes.
Mhm.
And we need that to properly adjust the tissues that I just mentioned, like the adipocytes and the liver, etc., to control and maintain the glucose levels as constant as possible.
Mhm. Yes,
Otherwise, I mean,
it's just a highly sensitive
maybe
control system.
yeah,
Um,
yeah,
so I want to ask you, I know that I think this ties into a lot of the other stuff that we're going to get into, but basically a big part of the pancreas, as we said, is an endocrine organ. One of the major hormones that is being secreted is insulin. That's what we just talked about. But there's probably a bunch of other sensitivities here to other hormones and other signaling pathways tied to other hormone systems. Can you start to talk about some of the other hormones that the pancreas is sensitive to? For example, I know that you've done a lot of research on things like estrogens. So, how do some of these other hormones fit into the picture when it comes to the pancreas?
yeah. I mean, there are different hormones that can control the insulin release. Before entering into estrogens, perhaps it's easy to think about when we are stressed and adrenaline is released, that what it does is to decrease insulin response to
Mm,
glucose so that we have more glucose in blood to go running,
yeah, yeah. If I
uh,
need to run away
because
from something scary,
we need it,
yeah, yeah.
yeah, if we are in danger and we have to run. So adrenaline, of course, affects the pancreatic beta cells in that sense. And later there are other hormones that may have a more long-lasting effect, such as estrogens, for instance, which are the female hormones. And we are interested in how particularly estrogens affect the pancreatic beta cells because the islet of Langerhans, the endocrine pancreas, adapts to pregnancy. It has
Mm,
to adapt to pregnancy. This is a very interesting period, pregnancy, to study from the physiological point of view, but very difficult as well. But during pregnancy, a woman has an increase in tissue because of the fetus, the placenta, and all
mhm.
that is growing. And the pancreas, her pancreas needs to adapt to that increase in tissue.
Hmm.
And so therefore, there's an increase in the number of pancreatic beta cells. There is also an increase in how cells senses glucose. They start sensing glucose to low levels and secreting more insulin. Uh, and in part, this is produced by estradiol and other hormones as well that are uh, particularly going up during pregnancy as placental lactogens,
Interesting, interesting. So,
for
so
instance.
yeah, this makes perfect sense. So obviously, when a woman gets pregnant, there's a there's a growing fetus inside of her womb.
Mhm.
She's going to need to provide nutrients and energy to the growing baby, and so it makes perfect sense that something like this system would have to change because it has to it has to adapt to that change in the woman. You're going to have to provide extra nutrition for the two bodies now instead of the one.
Yeah.
And if, to anticipate maybe some of the places we're going to go, it also makes perfect intuitive sense that something like an estrogen would regulate that process, because estrogens are naturally going to be fluctuating over the course of pregnancy. And so all the stuff is hooked together so that there's these metabolic adaptations in response to these hormonal changes triggered by pregnancy. But I would also imagine if estrogens instigate those changes in the pancreas during pregnancy, changes in estrogens in other contexts would probably naturally alter pancreatic function as well. So if we're, for example, living in an environment that is highly estrogenic because we're exposed to estrogens in the environment, that could potentially affect insulin signaling and pancreatic function in everyone, not just pregnant women.
Yeah, indeed, that was the idea when we started with our project using bisphenol A, which was known to be a xenoestrogen that was known to act as an estrogen, as a weak estrogen, the people said. That was 25 years ago, something like that. So the idea is, okay, if this is working like an estrogen is doing the same as the natural hormone to pancreatic beta cells in mice, so that we start doing is giving bisphenol A and estradiol, the natural hormone, separately to male mice. They never had to be exposed to these doses of an estrogenic compound, as you just mentioned. So, and then we discover something that was fascinating from the scientific point of view, but scary from the personal point of view too. And is that bisphenol A at low doses was producing insulin resistance and hyperinsulinemia in mice. And it was affecting insulin release and other peripheral tissues as well in adult mice. That was the first
So
study we did.
Basically, when we think about things like insulin resistance and type 2 diabetes, which we'll probably talk about in different ways, normally, I think a lot of people, when they think about why someone becomes diabetic, they think about, if it's type 2 diabetes, at least, that this is induced by the diet, that it has to do with sugar and carbohydrate intake. And indeed, those things are very important. But I think what you're starting to say is that you can get insulin resistance by other means. You could be exposed to certain hormones, for example, or certain endocrine disruptors. And, even in the absence of, say, drinking Coca-Cola every day, you can still develop insulin resistance in those ways.
Yeah, exactly. I mean, what I think we know now is that many of these endocrine disruptors that we study are part of the diet as well. For instance, bisphenol A, phthalates, and other bisphenols are part of our plastics components. And I mean, they are in food contact material. So when we buy something that is involved in plastic, so this plastic is going to release these bisphenols, phthalates, and other chemicals. Plastic contain hundreds or thousands of chemicals. Indeed, it's not only a polymer.
Yeah,
and I think this is part of our diet as well. So we always think in lipids, sugars, and of course, as you said, this is very important, and there's no doubt that they affect, insulin sensitivity, insulin release, and glucose homeostasis, and out of esogenic in general, the excess. But there are other chemicals in food coming from other sources,
Yeah. And for better or worse, these things are in our food supply, and to some extent they're unavoidable. A lot of the stuff is fascinating, but a lot of the stuff, the stuff you work on is fascinating, but it's also terrifying. So a lot of the stuff is going to be pretty unsettling, maybe, to people who haven't looked into how widespread these endocrine disruptors are. But before we really dive into some of that stuff, I want to talk a little bit about estrogen signaling and estrogen receptors and sensitivity in the body. So obviously, the body has natural estrogens that it produces. As you mentioned, we've got BPA and other xenoestrogens, artificial estrogens that we're going to talk about. Can you say a little bit about estrogen receptors? How many estrogen receptors are there? And is there anything sort of special or notable about them in terms of their sensitivity or how they work that's going to be important for understanding some of the other things we're going to talk about?
Yeah, there are three estrogen receptors. Two of them that are the more classical ones are estrogen receptor alpha and estrogen receptor beta.
Mm-hmm.
They belong to the so-called family of nuclear receptors, and they classically act as transcription factors.
Hmm.
So they bind the ligand, the natural hormone, in this case 17-beta estradiol. They dimerize, they go to the nucleus or are already in the nucleus, depending on the cell, and bind to an estrogen response element in the DNA, and that puts all the machinery of transcription to work, okay, and increases gene expression. And there is another type of response, which of these very same receptors, which is not in the nucleus. It's what we call extranuclear initiated effects. So in the last 30 years, there have been many different studies that have been published to show that these receptors, ER alpha and ER beta, are able to activate kinases outside the nucleus, such as PI3 kinase, ERK1/2, MAP kinases in general, and then to regulate other proteins or signaling pathways, including ion channels. So we have like two ways the cell respond to estrogens in terms of estrogen receptors. The nuclear one, which is the very classic one, and the extranuclear, which is rapidly initiated and activates as signaling. And this one is very particularly important in terms of endocrine disruptors.
I see. So oftentimes when we think about a receptor, they're often located on the outside of the cell, and the cell responds to something on the outside, and this leads to changes on the inside of the cell. But for for these estrogen receptors, they are internal receptors. They're either located in the cytosol or in the nucleus. And in the nucleus, when something like an estrogen binds these nuclear receptors, that's directly leading to transcriptional changes. So genes get turned on or turned off, things like that.
Yeah, exactly. And later, there is one more estrogen receptor that was proposed about 2005. And it's a G protein coupled receptor. It's called GPER. It's at the membrane. And it seems that it binds estradiol as well and have produce estrogenic signaling.
I see.
This one is less studied, but it seems that it's also there at the membrane. So we have
three layers of control.
Receptors, exactly, and one at the membrane. And they crosstalk, so that makes life complicated.
Yeah. So, estrogen is flowing through the bloodstream. It can get into cells. It can get all the way through the membrane into the cell, through another membrane, that's the nuclear membrane, and it's going to regulate gene transcription in the nucleus. It can also get through the outer membrane, but not pass through the nuclear membrane and regulate what certain proteins are doing in the cytoplasm. And there's also potentially these estrogen receptors on the outer membrane. And so you've got like three interacting levels of control inside individual cells.
Yeah, to the same hormone.
To the same hormone. Yeah. And is that special about estrogen, or is that sort of a normal thing when it comes to sex hormones generally?
It's now known that it's not only for estrogens. I would say for steroid hormones, we can say that, and there are the nuclear receptors act outside the nucleus as well for androgens, progesterone, vitamin D as well, and whether there is G protein coupled receptors, that's a different story.
Hmm.
has been proposed for some, uh, but not for other steroids. But
Yeah.
it is very well accepted, I would say, that nuclear receptors act as transcription factors in the nucleus and also outside the nucleus uh, for most steroids
Hmm.
hormone.
And are most tissues in the body sensitive to estrogen to some extent, or is it highly concentrated in a specific subset of tissues?
I would say that all tissues in our body are sensitive to estrogens.
Yeah, yeah. And
Yeah,
I mean, I
yeah,
guess that makes
in
sense.
general.
Yeah,
Yeah.
I guess that makes sense because, if you think about something like just the sex hormones in general, they are inside of everyone. They fluctuate across the lifespan. If you think
Mhm.
about something like puberty, right, the whole point of puberty is you need to transform all of the tissues in the body in a certain way. And so I guess these hormones are important for initiating that transformation. And it makes sense that, more or less every tissue is going to be sensitive to some extent.
Yeah, yeah, but I think it makes sense. And perhaps there is some tissue that it doesn't have some estrogen receptor or estrogen receptors, but I'm not aware of it.
Mm-hmm, mm-hmm,
It is true that we have these three receptors, the two nuclear and the GPCR, and it is true that each tissue may have different level of expression of
yeah,
each
yeah.
receptor. That is true. But as far as I know, most tissues express estrogen receptors.
Mm-hmm. And I want to start to talk about, endocrine disruption a little bit. I don't know if there's anything you want to set the stage with, but I suppose an endocrine disruptor would just be any exogenous molecule that can affect hormone signaling in an unnatural way. Is that a fair basic definition?
Yes, this I would say is the definition of the endocrine society. Indeed, is uh, an exogenous chemical, which can be natural or uh, or not,
Mm-hmm.
or artificial, but it affects hormone action.
Mm-hmm.
That's the more extensive definition of endocrine disruptor.
Yeah. And so how did you get into this field, and why has this become such an important area of study?
Yeah, when I started, really was at the end of the 90s when I came to Spain back from England, from London after my postdoc, and I had to start my own research line. So I was starting. Really, my question was how estradiol has a role in adapting the pancreatic beta cell to pregnancy.
Mm-hmm.
So then I was at home with my wife and we were watching TV and we watched a TV documentary that was named Assault on the Maid. It was a BBC Horizon program
Hmm.
and one of these documentary and most of the pioneers in the field of endocrine disruptors appeared there.
Hmm.
And they were explaining, how these endocrine or these chemicals were acting through nuclear receptors to affect different tissues. Of course, nothing about uh, obesity or diabetes at that
Mm-hmm.
time. And because we were using in the lab estradiol and see the adaptation of beta cell using estradiol, say why we don't start measuring what happened with this compound called BPA, that
Hmm.
is bisphenol A, that at that time was not so famous as it is now. That was in 97, 98. So I tested measuring ion channels, measuring the KTP uh, channel activity with patch clamp technique. So I acutely added bisphenol A at a very low concentration, 1 nanomolar, and 5 minutes after uh, perfusing the cells with bisphenol A, the channel closes.
Hmm.
Uh,
So a tiny
and
amount
I
of
said,
BPA had a big impact.
tiny amount. This is the amount we have in blood.
Oh, this is
And
this is
it's
the amount that's in our bloodstream
yeah,
potentially right
yeah,
now.
it is between 1 and 18 nanomolar
Wow.
BPA. That has been measured, and is more or less the amount of BPA we may have in blood.
Hmm.
And yeah, and then we started. You know, that's
Yeah.
how
So
we.
you saw this documentary and that made you want to do an experiment with BPA, and then you got started down this whole path.
Yeah, yeah, that was it. So yeah,
What let's explain for people very explicitly what is BPA and where does it come from most commonly?
yeah. Bisphenol A is one of the main is the main component of polycarbonate plastic.
Hmm.
So polycarbonate plastic, which is normally the transparent plastic that we have is hard. For instance, these glasses, this is no glass anymore, is polycarbonate.
Hmm.
So this is made by bisphenol A, and this is the polymer. And it's in many epoxy resins. It's in the interior of canned food. I mean, there is a plastic in there
There's a
in
plastic
contact.
liner in
Uh,
metal
uh,
canned foods. So most people probably don't even realize that.
exactly. Yeah, it is made. It contains bisphenol A, and bisphenol A is used also as a plasticizer that makes the plastics softer, so you can give a shape to the plastic, etc. Otherwise,
Hmm.
some of the polymers are very hard, and it's difficult to give a shape, and you need to solve the plastic. So BPA is added for that.
Hmm.
So BPA is practically in any plastic in a way or another. It's in PVC,
It's
yeah,
all over.
all over, all over. That's one of the problems. These, indeed, I wanted to be, these are recent results from a big European consortium uh, that is called Human Biomonitoring for EU. Um, they released all the data in the last year. You know, they have been studying the levels of chemicals in the whole European population. And the conclusion is that all Europeans above 18 have BPA in their blood
Hmm.
uh, at low doses, at least.
Yeah. But you said
That means
earlier that,
about 1 nanomolar, you
yeah.
know.
So is it safe to assume that virtually everyone listening to this has some amount of BPA in their bloodstream, and it's even if it's a quote unquote small amount, it's probably a physiologically active amount?
I would say so. Unfortunately, I would say that more than 90% of the people, uh, they at least in the US
Hmm.
and in Europe, where it has been measured, um, yeah, have BPA in blood, probably at a level that affects cells.
Yeah. So this isn't necessarily a thing where BPA is bad, but you need a very high dose of it, and trace amounts, are probably safe. This is a thing where potentially even low amounts could be problematic or could be having some physiological impact.
Exactly. That we see is that low amounts affect cells,
Yeah.
in
Yeah.
particular insulin releasing cells. So
Yeah. And I want to dig into that a little bit. So, from your studies and from what's known in the field, what is BPA? What kind of effects is BPA having with respect to insulin signaling?
yeah, with BPA, what it does is to first we can divide in two different uh, set of times. Let's say acute effect. So if we apply BPA, we very rapidly increase insulin release
Hmm.
in a matter of minutes. And this has been studied in isolated beta cells from mice, from human as well. And we know that it's mediated through the closure of the KTP channel. And it has been also demonstrated in humans by drinking just one dose of BPA, that affects um, insulin release.
Yeah. So within
And
minutes, it's already doing something.
within minutes, it's already doing something. Yeah.
Hmm.
And later, we have done experiments more long lasting, let's say in vitro, in culture cells during 48 hours, exposure to 1 nanomolar BPA. So they regulate genes, they regulate insulin gene expression, for instance, and they change the content of the insulin, the pancreatic beta cells, increases the content of beta cells. They regulate potassium channels, calcium channels, and sodium channel expression at the membrane, and also the function of those channels. And and this is associated with an increase in insulin release.
Hmm.
So what BPA does is to produce an oversecretion of insulin.
I see. So it's not only immediately affecting the release of insulin, it's also having longer lasting changes that increase the beta cell's capacity to release insulin.
Exactly. And this is not only in experiments in vitro that are very good for understanding the mechanistic insights, but also in mice. I mean, if we inject with BPA or we give oral BPA to mice, they get hyperinsulinemia, they get insulin resistance, glucose intolerance, uh, high levels of free fatty acids, high levels of leptin as well, etc. in adults, males.
Mm-hmm.
Uh, yeah, and then there are other studies done during pregnancy, so we can talk later about that as well,
Yeah, but I
if
guess
you
on a
wish.
basic level here, BPA is having a sort of pro-insulin effect. It's causing more insulin release. It's doing that
Yes.
immediately, and it's also leading to long-lasting cellular changes, such that the cells are basically able to release more insulin more often. And this is obviously going to tend to make it easier to develop things like hyperinsulinemia and insulin resistance. And if I can anticipate where this might go, um, this would suggest that let's say my great great grandparents,
Mm.
they were fairly metabolically healthy. They didn't have insulin resistance and things like that. Even if I was eating the same, someone today could be eating the same diet, the same nutrients in terms of fat, carbs, and proteins as, say, their great great grandparents. But if their great great grandparents were not exposed to BPAs and other endocrine disruptors, but you were living today, you might develop something like insulin resistance and diabetes in response to the exact same diet because your body is now more predisposed to do it.
Yes, I would say that's correct. Indeed. I mean, we have some chemicals. BPA is one example. There are others uh, that affect insulin sensitivity in peripheral tissue, including the skeletal muscle, the liver, and the adipocytes,
Mm-hmm.
and affect insulin content and insulin release.
Mm-hmm.
So are affecting your glucose homeostasis, and then in response to even the same diet as your grandparents, you may have a different control of glucose in your blood.
Yeah.
That's true. So
Yeah. Wow. How so when things like type 2 diabetes and insulin resistance are starting to develop, and even when they've developed, considerably, how easy is it for the body to reverse that? If you remove things like BPAs, if you change the diet, like the glucose and the carbohydrate intake, are these states, hyperinsulinemia, insulin resistance, things like this, are they reversible?
yes, it depends on the time. I mean, it's a very complex issue. Perhaps we have simplified a little bit, just to make it understandable,
Mm-hmm.
to be able to understand. But yeah, I think the point is for how long you are having those changes,
Yeah.
and depending on the time that you are you have the decrease in insulin sensitivity or the pancreatic beta cells overworking or whatever, it depends of how long that it can be reversible or not. So, for instance, we have done with adult mice, if we inject bisphenol A during 4 to 7 days, so a week,
Hmm.
we get the metabolic changes. And later, we just don't inject for 4 or 5 days, and everything is reversible
I see.
in adults.
In adults.
In adults,
So at least at the early stages, it's fairly readily reversible if it's
yeah,
just starting.
yeah. And this is something that happens as well with, for instance, people that is overweight or obese,
Hmm.
and they have a decrease in insulin sensitivity independently of BPA and the hyperinsulinemia. If they lose weight, even less than 10% of weight loss improve the metabolic parameters very
I see.
much.
So
And
even something like a less than 10% weight loss can lead
yeah,
to better
so
insulin sensitivity.
yeah, and if you start to exercise and to take care of this situation a little bit, you can improve. Uh, so yeah, in the case of BPA in adults, it is reversible. But it's something different in the case that during pregnancy you get an exposure to endocrine disruptors such as BPA or others.
Mm,
During pregnancy, it will affect the fetus and the mothers. And then the effects are long lasting for the
I see.
fetus and for the mothers as well,
So
for
basically,
dams.
yeah, if a pregnant woman is exposed to certain endocrine disruptors at certain phases of development, I would imagine, there's certain sensitive periods. This can basically lead to permanent changes that will be, lifelong lasting for the baby that's born.
Exactly, exactly. So that's, I mean, uh, the fetal development is when most genes are, uh, expressing and stopping expression and
Yeah,
all
yeah.
this kind of stuff. And then is the most susceptible period for this kind of endocrine disruptors acting through nuclear receptors, you know,
Hmm.
uh, to make epigenetic changes and then altering the
Hmm.
uh, you, you know, uh, uh, gene expression later in
Yeah.
life as well, altering the phenotype later in life.
Does BPA or other endocrine disruptors, do they actually cross the placenta and get directly into the fetal bloodstream?
Yes, indeed, BPA does. And others, such as dioxins, they do as well. Most of the lipophilic uh, endocrine disruptors are able to do that. So
Hmm.
they pass the placental barrier and go to the fetus and signal there.
Hmm. And I think you said that. So not only can it lead to um, permanent, long-lasting changes for the developing fetus that will go all the way into adulthood. I think
Mm.
you said also that it can lead to long-lasting and permanent changes for the mother. So the mother's own endocrine system can be disrupted such that it sort of doesn't go back to normal or back to baseline after the pregnancy ends.
Yes, I mean, this is less known that the effects on the fetus, but for instance, during pregnancy, if mice are exposed to endocrine disruptors, so we did it first with BPA, we already published that in 2010, and later have been done with dioxin as well. Um, then you get a change in the hormones in the mother
Hmm.
during pregnancy. So it's not only passing the placental barrier and going to the fetus, it's that it's producing an, a change in insulin levels in plasma, leptin levels in plasma, and lipids in the mother. And that changes the environment for the fetus.
Yeah.
So it's not only the endocrine disruptors passing, but the whole environment in the mother is changing. And later, if you study those mothers as we did, everything goes back to normal during 3 4 months. And after that, the mothers started to be overweight, insulin resistance, glucose intolerance. They have decreasing beta cell mass, decreasing insulin release from beta cells, and all kind of metabolic alterations in dams, you know.
Yeah. Wow.
And this is pretty new. It needs to be very much studied, but I think it's like a new uh, sensitive window for the mother.
For the mother.
I mean, yeah, pregnancy is a very
Yeah.
plastic period for the
Yeah.
mother as
Yeah.
well. And I mean, we have a project now uh, that we're going to try to understand what mechanisms are taking place in the mother to have these long-lasting effects that
Yeah. Yeah.
appear later. Even they disappear and they appear for months afterwards.
Yeah, yeah. And immediately you wonder, it's difficult not to wonder about common experiences that certain women have with pregnancy. So, for example, some women have a child, one or two or three children, say, and they have no trouble or very little trouble losing weight and getting back to where they were before the pregnancy. And other
Mhm.
women struggle with weight gain that doesn't go away and things like this. And one wonders if this ties into that, if it has something to do with exposure to things like endocrine disruptors like BPA.
Yeah, of course, this is pure speculation, but I mean, what I can say, our experiments is that mice that were exposed to BPA during pregnancy, uh, later in life, they were overweight, had these metabolic problems. And we did a control, just exposing for the same time non-pregnant females.
Yeah.
We exposed to BPA exactly the same time, and they had no overweight and no metabolic problems.
Yeah.
This is what I can say. I don't know if that, I mean, we need to
Yeah. We don't know if
do
that's true in
studies,
humans for sure, but
exactly,
yeah.
but it's a possibility, at
Yeah.
least that
But
we,
basically what you're
we
saying
need to
is,
think about.
yeah. So pregnancy is basically, and we're talking about mice here, but, by analogy, it makes sense that this would apply to other mammals. Pregnancy is sort of like it's a critical period in the adult mother. Normally, we think about critical periods of development. We think about childhood and infancy and things like this. But in the
Mhm.
adult mother, things are basically more plastic while she's pregnant because obviously her whole body needed to be plastic, needed to rearrange itself in
Mhm.
order to accommodate the fetus. And so this can open up the opportunity for long lasting changes to happen in a pregnant female animal that would not otherwise happen to a non pregnant adult female animal.
Yeah, exactly. That's a very good summary indeed of what
Wow.
I try to say. Yeah,
It's yeah, I mean, it's just it's fascinating. It's simultaneously fascinating, but obviously very unsettling.
yeah, it is scary. I mean, it's always you have this mix of feeling, when I see
Yeah,
those
yeah.
results. That's true.
And I want to ask you about sort of the, you've already said that BPA is pretty potent. Can you say a little bit more about like the dose and the potency of BPAs and other xenoestrogens compared to natural estrogens? Are these things generally more potent than natural estrogens, less potent? How do you start to think about that
Hmm.
stuff?
Yeah, I think that's a very important question actually, and one of the main problems that I think we need to still to understand.
Mm-hmm.
Let's say, for instance, bisphenol A and other bisphenols, they have a low affinity for estrogen receptors,
Mm-hmm.
about 500 to 1,000 fold less than estradiol.
Got it.
Okay, but uh, they act through these receptors at doses at the same level as estradiol. So let's say the affinity for bisphenol A of estrogen receptor is within the micromolar range.
Yep.
But we have effects, we and others, we have effects at 100 picomolar, 1 nanomolar,
I see.
which is far below the affinity of the receptor for
Uh, so it's
the
acting
ligand,
by it's acting
for
through
the
other
BPA.
means.
Well, I mean, we are working on that, and yeah, we are trying
Mm-hmm.
to publish a paper in a good journal now. And that we found is that mainly bisphenol A is acting through estrogen receptors outside the nucleus. Nuclear
Mm,
receptor, but through the extranuclear initiated pathway.
I see.
And when they do that, they have a very high efficacy
Ah,
because there are ways to amplify the signal. That's one thing.
hmm.
And the other is that there is a, a proton transfer from the ligand. This is what we're trying to publish is some new results now to uh, activate, to switch this extranuclear pathway.
Hmm.
So it's like having a kind of different mechanism to activate this extranuclear pathway compared to the nuclear pathway.
I see.
So the
So,
nuclear pathway is uh, normally has to work with a high affinity
yeah,
ligand,
yeah,
and outside the nucleus, it can work with a low affinity ligand,
I see. So
being with a high efficacy.
so the basic idea here, if I'm hearing you correctly, is so we said before you've got these three levels of control in terms of the estrogen receptor. You've got intranuclear receptors in the nucleus, you've got
Mm-hmm.
receptors in the cytoplasm on the inside of the outer cell membrane, and then you've got
Mm-hmm.
receptors on the outer cell membrane. Even though these BPAs might have a lower affinity, a lower stickiness for the receptors than natural estrogens, because you've got these three levels of control, these three different types, these three different flavors of estrogen receptors, the BPAs might be acting very, very potently at that outer layer so that they can have strong effects even at a low dose, even though their affinity for the receptor is actually less than the natural estrogens.
Yeah, exactly. It is easier. Well, it is easier if the receptor is outside the nucleus,
Yeah.
interacting with other proteins such as kinases, and then produces second messengers. The signal gets amplified,
Amplified. Yeah.
and this is something that it doesn't happen in the nucleus.
Hmm.
Uh,
I
because
see.
yeah, there of course is one ligand, one receptor, and one estrogen response element in the DNA. So
Yeah.
there is no amplification in that sense. But outside the nucleus, you can interact and produce, for instance, cyclic AMP and produce many cyclic AMP molecules with just one ligand, and then have many PKA
Yeah. Yeah.
activation and
I see.
phosphorylation, etc. So the signal gets amplified. And in
Yeah.
addition to that, so we think that there is another switch to activate these extranuclear effects, at least with BPA, that is more kind of a chemical reaction that's only the shape of the molecule inside.
I see. Yeah. So it's like at these different layers of cellular regulation, the signal can get amplified to different extents. It's sort of like,
Yep.
so you said the nuclear receptors. It's more like a one-to-one thing. It's like you and I talking here. I could ask you to do me a
Yeah.
favor, and I'm just asking Angel to do me a favor. It's one
Yeah.
person. But if I go to my balcony and I shout to a group of 10 people, I can now amplify my signal tenfold. And that's sort of like the outer layer of signaling here.
That's a perfect example. Thank you. Yeah.
Okay, but the point is basically, because there's this sort of question that I think some people have that I've had when I think about some of this stuff. It's like, okay, we've got all these estrogen receptor, these estrogen, exogenous estrogens in the environment. The environment seems to be net estrogenic. It's, when we look at sort of coarse grained signals in men and women, both of them seem to be drifting in the estrogenic direction. But the question in my mind was, well, okay, if we've got these, estrogens, but they're actually less sensitive, they're less potent, or they have less affinity for the estrogen receptor than natural estrogens, they might have a net anti-estrogenic effect if they're sort of pushing the natural estrogens out of the way and they're less potent. But basically, what you're saying is that, at least in the case of BPA, because of signal amplification and things like this, they can have a net estrogenic effect, and that makes sense.
Yeah, it may have an estrogenic effect, but it not always does exactly the same as the natural hormone.
Mm,
that's another point.
Yeah. It's not having the
Uh,
exact same set of
no,
effects
no,
as the natural
not at
hormone.
all. No,
Yeah.
no, no, indeed, because well, the receptors they need to dimerize to produce, for instance, and you have homodimers of estrogen receptor alpha, homodimers of estrogen receptor beta, and heterodimers estrogen receptor alpha and beta. Okay, and the ligands can make a different pool of these dimers. So we have studied that, and it seems that bisphenol A doesn't produce heterodimers, but estradiol produces heterodimers of ER alpha and ER beta. So because of the pools of dimers is different between estradiol and BPA, that may be a reason to have different signaling. So some responses are going to be equal
Yep.
between both ligands, and some are going to be different.
I see. So I think
Because it's not
yeah.
exactly the same, and that's
Yeah.
an important point.
Yeah, that is important. So the exposure to these xenoestrogens, it's not a quantitative change in estrogen signaling. It's not just sort of boosting up or down natural estrogen signaling, period. It's a different pattern of signaling and stuff happening in the cell that's
Yeah,
not just a different amount of estrogen signaling.
yeah, exactly. This is I how I look it personally is okay. Really, what BPA and other xenoestrogens is doing is altering, disrupting the estrogen signaling
Yeah.
somehow. So it's not really mimicking or antagonizing completely, but is
Yeah, it's truly a disruption.
it's a disruption. It's just making a different signal and then modifying the signal for the natural hormone because
Yeah.
BPA is messing around.
Yeah. Um, so obviously, when we think about it makes sense that we're talking about estrogen signaling in the context of things like pregnancy and early development. But of course, all of these hormones, estrogens, androgens, everything's in males and females, just, it's in different amounts. There's different patterns of fluctuation at different periods of the life cycle. We talked about pregnancy and fetal development a little bit, but I want to think about how these things can affect males versus females now. And it's natural to imagine that the effects would be quite different because the pattern of estrogen receptors and other things is going to be distinct in males versus females. What do we know about effects in males versus females for these estrogens like BPA?
Hmm. Uh, there is sex differences. This is true. I would say for BPA and yeah, the experiment that have been done in the past uh, in terms of glucose homeostasis or obesity, uh, I think the effect, at least with BPA, is more in males than in females. It seems that females are protected, but the studies were not really designed to study different in sex. But new studies have been done, for instance, with dioxins, and it is not a xenoestrogen, but it's acting through the aryl hydrocarbon receptor, which is another nuclear receptor. And these are results from Jennifer Bruin laboratory in Carleton University in Canada. And they show very well how the phenotype in terms of glucose homeostasis is different between uh, males and females. And they designed the study to follow males and females. And it seems that in the case of dioxins, uh, the action is mostly in females. They have a more dramatic phenotype that males
Yeah.
actually.
And I guess
Yeah,
I
yeah.
guess on a very basic fundamental level, we would expect there to be a lot of sex differences when it comes to endocrine disruptors because the differences between the difference between a male and female comes from it has an endocrine origin, right? It's the
Yeah,
endocrine system is
yeah,
actually
yeah,
sculpting the bodies differently and making them
yeah,
differentially sensitive to all of these
yeah,
hormones. So we should expect that, I think, right?
yeah. Indeed. I mean, for instance, in our experiments, we have never seen that phenotype of insulin resistance, glucose intolerance, or hyperinsulinemia in females. So this is why we
In non-pregnant
started to do
females.
it in non-pregnant females, of course. I mean, in offsprings,
I see.
or in adults
I see.
where we treat
So,
it.
so you've got results where
And
BPA exposure in adult males and adult
males,
non-pregnant females,
yeah,
you get bad effects in the males that you don't see in the females so
we
much.
don't see in the females. We didn't publish in the females. We just didn't see anything.
Full version of this episode is available to paid subscribers at the Mind and Matter Substack at mindandmatter.substack.com. The full version is also freely available on my YouTube page. Just look up Mind and Matter or my name, Nick Jikomis, on YouTube, and you'll find the episode there. This is a partial version of the episode, and so it'll cut off partway through, but you'll still get a significant portion. So sign up to become a paid subscriber at Substack or visit my YouTube page to get the full episode. This episode is supported in part by Athletic Greens. Their main product, AG1, is a comprehensive and convenient daily nutrition product containing 75 vitamins, minerals, and whole food sourced ingredients with less than 1 gram of sugar per serving, no nasty chemicals or artificial anything. It's gluten and dairy free and compatible with paleo, vegan, vegetarian, and ketogenic diets. AG1 is a quick and convenient way to supplement your diet to help ensure your body is getting the nutrients it needs. It comes in powder form, and you can mix it in water and drink it, or you can put it into a smoothie or a shake or something like that. I mix it into water and drink it, and it's super convenient. If you go to athleticgreens.com/mindandmatter, Athletic Greens will give you a free 1-year supply of vitamin D and 5 free travel packs with your first purchase. Their vitamin D product comes in tincture form, so you just take 1 drop each day. A large fraction of the population is actually vitamin D deficient, especially in winter months when we get less sun exposure, and vitamin D is super important for the proper function of the immune system and for a variety of other things. So visit athleticgreens.com/mindandmatter or click the link in the episode description. You'll get a free 1-year supply of vitamin D with your first purchase.